Hey everyone, Dr.Mungli here. So, in this video, I will be explaining you what exactly is xenobiotics metabolism and what is the biotransformation of xenobiotic compounds in our body. So, now let us get into know what exactly is xenobiotics. So, xenobiotics, these are the chemicals, these are the substances which get into our body in the as a component of our food, or maybe they are food additives, or these are the drugs, these are the chemicals, and these are the pollutants, carcinogens, all these are put under xenobiotics. So, as such xenobiotics that they do not have any nutritive value and they are not required for our body under normal circumstances, and rather they are potentially harmful to us. So, where exactly xenobiotics metabolism will go on, and what exactly the properties of xenobiotics here. Xenobiotics, these are mostly lipophilic molecules, that means these are hydrophobic molecules and they cannot be soluble in water. So, in a xenobiotic metabolism what happens is these hydrophobic or lipid soluble molecules are converted into hydrophilic molecules, so that they can be excreted out of our body. In the urine it can be excreted in the body through feces, or it can be going out of our body in the sweat. So, the major organ in our body that handles these xenobiotics is the liver. Liver is the principal organ where xenobiotics metabolism is going on. So, other than the liver, so there are other organs that can be involved in xenobiotics metabolism, and that is it can be happening in the small intestine or it can happen in the kidney. So, now let us see like how exactly liver is handling this xenobiotics. So, the xenobiotics are generally metabolized in the liver. This is the principal organ in our body. So, the metabolism of xenobiotics involves two phase reactions. One is like phase 1 reactions and phase 2 reactions. So, what exactly happens in the phase 1 reactions and what exactly happens in the phase 2 reaction. So, in the phase 1 reaction, so I am showing it here in the figure. So, xenobiotics are hydrophobic molecules, they are not so they are lipophilic, they are not soluble in water. So, I am mentioning that as RH here. And phase 1 reaction generally it involves cytochrome P450 enzyme. So, the cytochrome P450 enzymes. So, basically in this phase 1 reaction what happens, there can be reduction of these substances, there can be oxidation of substances, it can be introduction of hydroxyl group, or there may be hydrolysis of these molecules. So, the phase 1 reactions most commonly involve cytochrome P450 enzymes. Now, in the phase 2, so with this what happens, so there will be an hydrophilic molecule is introduced like mostly by cytochrome P450s. So, ROH hydroxyl group is introduced into this hydrophobic molecule. RH is now converted into ROH. Okay, now this is the primary metabolite that came out of xenobiotic. So, what happens to this ROH containing primary metabolite? This primary metabolite now it will get into phase 2 reactions. In the phase 2 reactions, so we have conjugation reactions where a molecule will be conjugated with some hydrophilic molecule which contains a negative charge, and it will be added to that, or it can be sulfation, addition of sulfur group like SO4 minus, or it can be methylation, it can be addition of glucoronidation. So, all these are the phase 2 reactions which will make secondary metabolite which are water soluble, they are ready for excretion. So, like example it is given in the figure is RSO4, like sulfate is added. So, with this secondary metabolite being soluble in water, it can be excreted out of our body through the kidney into the urine, or it can go out of our body through the intestine into the feces. So, and also it can go out of our body by the sweat glands into the sweat. So, this is how the xenobiotic metabolism is actually handled in the liver by phase 1 and phase 2 reactions. In phase 1 reactions most commonly reduction, oxidation, hydroxylation, hydrolysis can happen. Most common phase 1 reaction enzyme is cytochrome P450 class of enzymes. In phase 2 what happens, so the primary metabolite is further added with the negatively charged molecules, making it water soluble. So, in phase 2 reaction generally there will be conjugation process, or it can be sulfation, or it can be glucoronidation. Now, let us move on to see what all the different enzymes that are involved in biotransformation of xenobiotics. So, cytochrome P450 monooxygenase enzyme comes first. This is the most common enzyme which is involved in xenobiotic metabolism. Other than that, we have glutathione s-transferase enzyme adding like glutathione there, so thereby facilitating in the excretory mechanism. Dehydrogenases, we have reductases, then we have UDP glucuronyl transferases adding UDP glucuronate molecule, we have methyl transferases, we have sulfotransferases which will add sulfur, sulfate and mixed function oxidase, and UDP glucosyl transferases and acetyl transferases. So, these are the like amidases, esterases. So, these are the list of enzymes which are involved in biotransformation of xenobiotics. So, the most common one, the most important one here is the cytochrome P450 is glutathione S-transferase, UDP glucuronyl transferase and sulfotransferases. These are the things that I will be going over in detail as I move on. So, let us look at cytochrome P450 monooxygenase system enzymes. So, this cytochrome P450 monooxygenase enzymes, these are flavin containing heme containing molecule. They contain flavin which is a derivative of riboflavin, they contain heme, part of its molecule is heme with a ferrous iron there, and with the heme ferrous iron has got a maximal spectral absorbance at like 450 nanometer. That is why these enzymes are referred as cytochrome P450, P for pigment, 450 is the spectral absorbance 450 nanometer. And also cytochrome P450 monooxygenase system enzymes, they use NADPH plus H plus and also as a reducing molecule to reduce oxygen, molecule of oxygen. So, the mechanism is shown here, NADPH plus when it is oxidized to NADP. So, the FAD present in the cytochrome P450 is converted to FADH2 and FADH2 further reduces FMN into FMNH2 and that further keeps ferrous iron reduced in the heme of cytochrome P450. And then what happens? The RH which is there, this RH which is hydrophobic molecule, lipophilic molecule is introduced with an atom of oxygen to make ROH. That is how an atom of oxygen is used here, and the another atom of oxygen will be released as water molecule. So, here basically RH is introduced, basically the oxidation is going on here, RH is converted to ROH, like introduction of oxygen atom is done here, another atom goes out as water molecule and H2O, 2 protons will come from NADPH plus H plus there. So, that is how the cytochrome P450 monooxygenase system enzymes will work. Now, let us look into the general mechanism, general principles, general properties of cytochrome P450 monooxygenase enzymes. All of these enzymes causes oxidative, peroxidative, reductive degradation of exogenous or endogenous substances in our body. And also they are all found in smooth endoplasmic reticulum and are sometimes they are referred as microsomal enzymes. And also human cytochrome P450, a family of enzymes, there are 57 genes that can code for all these subclasses of cytochrome P450 and all of them share 40% sequence homology in their amino acid sequences. And also note that these are the enzymes have different but overlapping substrate specificities. So, that means there are a lot of subfamilies of cytochrome P450 enzymes, they have their specificities, but they can be overlapping specificities also. And one of the greatest, one of the most important cytochrome P450 which handles greatest number of drugs, chemicals is cytochrome P450 3A4, right? CYP is cytochrome P450, 3 is the subfamily, A is the substrate and 4 is the isoform of in that particular class. So, cytochrome P450 3A4, remember this is the isoform which accounts for 30 to 40% of cytochrome P450 enzymes in the liver and 70% of cytochrome P450 enzymes in the intestine. So, remember this enzyme, cytochrome P450 3A4 handles greatest number of drugs, chemicals and substances in the liver and most of it is in the intestine. Alright, so this is how like cytochrome P450 3A4 enzymes as enzyme as I said it handles greatest number of drugs in humans And also note that the drug metabolism by cytochrome P3A4 is based on the affinity of this substrate for this particular enzyme. That means there are a lot of drugs, there are so many drugs which are handled by cytochrome P453A4 enzyme. That means say if multiple drugs are taken at the same time, so at that time whichever the drug which has got higher affinity for this enzyme, it will be handled first and the other drug can actually it will be handled later. So, this means what happens when the multiple drug combination is given. So, one drug is preferentially metabolized than the other drug. So, other drug blood levels can increase, it can reach to toxic levels also if we do not really take care of that. So, we got to be very careful when the different drug formulation is done, multiple drug combination is given. So, there can be changes in the xenobiotics of these drugs. So, one drug preferentially metabolized than the other drug concentration can build up in the blood and it can lead to toxic levels or it can lead to side effects or the adverse effect. And also note that many substances or drugs can impair the metabolism of cytochrome P4 metabolism by cytochrome P453A4 CA43A4 can the metabolism can be catalyzing process can be inhibited by certain things, certain drugs, certain molecules. Example that is given here is grapefruit juice. This grapefruit juice actually it has got inhibitory effect on CYP3A4. So, and also note that CYP3A4 enzyme is involving in HMG-CoA reductase drug metabolism. So, whenever a person has taken grapefruit juice and HMG-CoA reductase inhibitors like statins, so there can be inhibitory effect of cytochrome grape juice on CYP3A4 which is actually metabolizing HMG-CoA reductase inhibitors. So, their drug HMG-CoA reductase inhibitor drug levels can be increased. It can lead to toxic levels, it can lead to adverse effects because of that. That is why some of the people who are taking grapefruit juice and also take HMG-CoA reductase inhibitors. So, their HMG-CoA reductase inhibitor drugs level can go up. It can lead to toxic manifestations like it can lead to muscle toxicity or liver toxicity, something like that. So, remember about it. And also cytochrome P450-2E1 CYP2E1, this is the enzyme cytochrome P450 which metabolizes alcohol. It is an alcohol metabolizing enzyme. This is also referred as microsomal ethanol oxidizing system. So, CYP2E1 is an alcohol metabolizing enzyme. And also note that the cytochrome P450 enzymes are induced by specific substrates, like some of the best substrates that these enzymes have. These are the substrates that they will induce the enzyme levels, thereby more and more enzyme levels will be elevated. Example like CYP2E1 which is a metabolizing alcohol. So, alcohol itself will induce CYP2E1 enzyme, thereby more and more CYP2E1 enzymes are available in the liver. This is what happens in alcohol abusers, because the alcohol abusers takes alcohol continuously, that means their CYP2E1 levels will be elevated and thereby they will have more CYP2E1 to metabolize alcohol. So, this can lead to certain complications later also. And also CYP2E1 cytochrome P450 is as such as a part of their metabolism. So, they are going to produce reactive oxygen species, or they are going to produce free radicals, and these free radicals can cause damage to our cell membranes, it can lead to cellular damage. So, that is why it is always good to avoid xenobiotics as much as possible by taking a natural food, or not really having too much of additives, food additives, preserving agents or chemicals, medications, drugs, all these things, pollutants, carcinogens, all these things need to be avoided. Now, let us see some of the examples for cytochrome P450 biotransformation reactions. First example that I am explaining here is a vinyl chloride. This vinyl chloride is used in the synthesis of plastic. What happens is the people who are exposed to vinyl chloride, if it gets into our body, so vinyl chloride is metabolized by the phase 1 xenobiotic metabolism, where CYP2E1 cytochrome P450-2E1 is going to convert vinyl chloride into chloroethylene oxide. This chloroethylene oxide further it will undergo phase 2 reaction into chloroacetaldehyde, and chloroacetaldehyde will be handled by glutathione S-transferase, where GSH is used and it is conjugated with the glutathione and may excreted out of our body. So, this is how it is excreted, but what happens, this chloroethylene oxide, if it is not going sufficiently into phase 2 reaction, as you can see here, this is a reactive epoxide molecule. Chloroethylene oxide is a reactive epoxide molecule, and it can covalently, it can react with the guanine in the DNA, it can covalently bind with the proteins and cause cellular damage, and this could cause angiosarcoma in liver of exposed workers, like those are exposed to vinyl chloride. So, they are tend to develop angiosarcoma in the liver because of the chloroethylene oxide, which is actually metabolic product of vinyl chloride done by cytochrome P450 This is what is a biotransformation. So, not always cytochrome P450s are involved in detoxification process. Sometimes their metabolic intermediate can also cause toxicity. So, this is why it is called biotransformation. Xenobiotics are undergoing biotransformation. Mostly they undergo detoxification, but some of them can lead to, some of the intermediates can lead to toxic effects. So, that is the toxification. Now, aflatoxin B1 is another example that I would like to explain here. It is produced by fungus Aspergillus flavus that grows on peanuts under damp conditions. So, what happens is cytochrome P450 enzyme, it biotransfers aflatoxin B1 into 8,9 epoxide, which is more toxic than cytochrome than aflatoxin B1. So, here what happens? So, the aflatoxin B1, which is a less toxic molecule, is actually converted into more toxic form done by cytochrome P450 enzyme, right. So, this is the toxification, not just a detoxification by cytochrome P450. As I was explaining before, there is a toxification also. Cytochrome P450s can also be involved in toxification of less toxic molecules into more toxic molecules. The best example is aflatoxin B1 enzyme. So, aflatoxin B1, so which is converted into 8,9 epoxide form. So, and also this 8,9 epoxide form, what it does, it is going to modify DNA, where it is going to introduce guanine into basically replace, it causes the mutation where there is introduction of guanine in by thymine. So, G is replaced by thymine here, and especially on P53. These are tumor suppressor gene, P53 gene, and potentially leading to carcinogenesis in humans. And also this 8,9 epoxide can combine with lysine residue in a protein, and it can act as a hepatotoxin in the liver. Now, let us move on to see our final example, that is acetaminophen or paracetamol, which is traded as like which is trade name for that is Tylenol in the western countries. Acetaminophen or paracetamol. So, it is metabolized by sulfation, it is metabolized by glucoronidation most of the time. As I have given here in the structure, acetaminophen or paracetamol, it is there in the center there. As you can see, it can go on either side. On the one side, it can go to sulfotransferase reaction, sulfation, and it can be excreted out of our body in the kidney or urine, or it can go into UDP glucuronyl transferase enzyme, UDP glucoronidation, and the UDP glucoronidate molecule will be excreted by the kidney in the urine. So, this is how acetaminophen paracetamol is mostly handled like sulfation glucoronidation. Only less than 10% of acetaminophen under normal therapeutic range, it will go into cytochrome P450 mechanism into a molecule called NAPQI. This NAPQI is n-acetyl para-benzoquinone imine. So, n-acetyl para-benzoquinone imine. So, this is what is NAPQI. This NAPQI, only less than 10% of it is coming from the metabolism of acetaminophen by 2E1, cytochrome P450 2E1, because most of it is handled, 90% of it is handled by sulfation and glucoronidation. Only less than 10% is going into 2E1 and making NAPQI. So, what is this NAPQI? So, this NAPQI, it is a toxic and unstable intermediate that can cause cellular damage by, it will go and bind with the protein, it will go and bind with the DNA, it causes cellular damage, and thereby it leads to hepatocyte death. So what happens is, so NAPQI, so under normal therapeutic range, so whenever the therapeutic range of acetaminophen or paracetamol is given, so only less than 10% of NAPQI is made by 2E1 enzyme, because more like 90% or more than 90% is simply handled by sulfation on so as you can see in the figure, on the right side you have sulfation, on the left side you have glucoronidation. That will take care. So only less than 10% is coming through TE1 and NAPQI, which is a molecule that can lead to toxicity, but the thing is we have a mechanism to take care of it. So we have glutathione s-transferase, as you can see down there. Glutathione s-transferase, what it does, it is going to attach glutathione and make mercaptouric acid, and that mercaptouric acid goes out of our body through kidney into urine. So this is how we are going to handle less than 10% of NAPQI that is coming from 2E1 enzyme. So remember, acetaminophen and paracetamol, as such they are not the toxic molecule, it is converted into a toxic molecule by 2E1. So that is a toxification. Cytochrome P450 2E1 is actually causing toxification, toxic molecule NAPQI, but fortunately we have glutathione s-transferase which is using glutathione and it is taking care of it. Now what happens, whenever a toxic doses of acetaminophen or paracetamol is given, or when there is acetaminophen or paracetamol toxicity, what happens? So the glucoronidation and sulfation, these two processes which are handling the most of the acetaminophen and paracetamol, they will be overwhelmed with the toxic concentration, because under normal therapeutic range and they will take care, but when there is excess they will become overwhelmed. Most of it is going into 2E1 now. So although 90% is taken there, but since the concentration is increased, 2E1 enzymes are converting more and more acetaminophen or paracetamol into NAPQI. And this NAPQI, although we have glutathione s-transferase, but there is a limitation for glutathione s-transferase. What it does? It needs GSH to do that. So when there is insufficiency of the GSH, when the GSH is not sufficiently available in the liver, so the glutathione s-transferase won't be able to take care of all the NAPQI that is made. When it exceeds more than 10%, so that means it does not take care of all the NAPQI, and this NAPQI, what it does, it will go and bind to the protein, it will go bind to the DNA, and it can cause hepatocellular death, and that is how it leads to hepatic toxicity. So the acetaminophen or paracetamol toxicity is not just because of that molecule, it is because of NAPQI which has got a toxic effect on the liver, especially when it exceeds more than 10%, that can be handled by GST. Now what is the treatment? So the treatment for acetaminophen or paracetamol toxicity is n-acetylcysteine. It is there in the figure down here. N-acetylcysteine, what it does? It is going to provide, it is helping in the restoration of GSH, because it provides cysteine, it is going to help in the regeneration of GSH. So GSH actually is synthesized, more and more GSH is synthesized, which will become part of, which will be used by glutathione s-transferase, and thereby liver can handle NAPQI and make mercaptouric acid. That is how an N-acetylcysteine can act as an antidote there by decreasing acetaminophen toxicity by handling NAPQI indirectly, because N-acetylcysteine is going to help maintain the concentration of GSH. GSH is going to be useful for GST, glutathione s-transferase, and that is helping in the conversion of NAPQI into mercaptouric acid. So that is how the antidote work here. Now, and also remember, alcoholics, as I said before, alcoholics, they are inducing this enzyme cytochrome P450 2E1, as you can see here. This is the enzyme that is involved in the conversion of acetaminophen into NAPQI. So when you have more of this enzyme, so the more of the acetaminophen can be converted into NAPQI, which can exceed the capacity of GST to take care of it. So this is what happens in alcohol abusers, because simply they have more 2E1, cytochrome P450 2E1, they will divert more of acetaminophen into NAPQI, and if they have less GSH there, they will be susceptible for toxic effect of acetaminophen and paracetamol. Remember this. That is why in alcohol abusers, acetaminophen and paracetamol prescription need to be monitored well, because there can be, because they are at high increased sensitivity, increased risk of toxic effects by these molecules, because simply they allow high levels of 2E1 enzyme. Okay. So that is about the figure there which I have explained. So in this slide I have written all the details all the like kind of notes for you, acetaminophen and paracetamol, how it is handled. So, it is metabolized in the liver by sulfation, glucoronidation, CYP2E1 for safe excretion by the kidney and urine. So, when acetaminophen is taken in correct therapeutic range, only less than 10% of it forms NAPQI, which is handled by GST by using GSH, that is glutathione s-transferase. So, when the dosage exceeds therapeutic range, sulfation, glucoronidation simply becomes, they will be overwhelmed by this, and that means more than 10% of NAPQI is made, which is formed by CYP2E1 enzyme, and the limited availability of GSH can impair GST activity, thereby building up more and more NAPQI that can lead to hepatocyte death. Alcoholics, they will have more cytochrome P52E1 enzyme, that means they are at higher susceptibility for converting acetaminophen into NAPQI, which can lead to hepatotoxicity in these people. Like n-acetylcysteine is an antidote for acetaminophen or paracetamol toxicity, because it is going to restore GSH, and GSH will have a good effect on GST, and GST will take care of NAPQI. So, this is all about xenobiotics metabolism, biotransformation of xenobiotics, what all the enzymes that are involved, what all the two types of reactions that are involved here, phase 1 and phase 2 reactions, and given example means I given details of cytochrome P450s, given examples like vinyl chloride, Aflatoxin B1 and acetaminophen or paracetamol. So, that is all about xenobiotics metabolism. I hope this video has really helped you in understanding this particular concept, which is commonly asked in the exams, certain like competitive exams also. So, I will see you in my next video. Till then, you take care.